Good day, friends, and welcome now back to the channel. Today we will discuss an interesting topic for microbiology. We dey call am gram staining. Now, gram staining na technique wey we dey use for microbiology to help us to identify different bacteria wey dey. You know say millions of bacteria dey, and the major problem wey microbiology dey get na how to identify all these bacteria. Now, this technique, this gram staining na technique, na one of the foundation technique wey we dey use to identify and differentiate bacteria. Now, the thing with the gram staining na say e get one scientist wey we dey call Christian Gram. Na for 1884 this man bin design this technique. Him bin design this method of how we dey use this method to fit differentiate different bacteria from one another. Now, this method e get many steps, and we go discuss those steps very quickly. I hope say you go understand them. E no dey difficult. You no need to cram am. You just need to understand wetin dey happen. Now, the first step na say you go prepare wetin we dey call smear. Now, that bacteria wey you wan identify, you need to put am for something wey we dey call glass slide. That glass slide na small small glass wey dey for laboratory. Now, if you don put that bacteria from whatever sample wey you collect the bacteria from, you go come put a drop of water. E get two ways wey you fit allow the bacteria. Either you go allow air to dry am, that one we dey call am air drying, or you go just run that slide over a flame very quickly. Short period, the thing don dry, the bacteria wey dey there don dey killed, and e don dey ready to carry out the actual gram staining itself. So if you don fix the bacteria, either you heat fix am or you air dry am to fix am, the next thing na to begin dey run all these processes. So the first one wey you go do na to add wetin we dey call the primary stain or crystal violet. Crystal violet na the name of the chemical wey we dey call the primary stain. Now, if you add am, pour am on top the glass slide where the bacteria dey, wetin go happen na say the crystal violet go dey react with components wey dey for inside the cell wall of the bacteria. So e go dey there, and that crystal violet, remember say the color na either violet or purple. Now, the second thing wey you go add na iodine. And iodine na wetin we dey call the mordant. And wetin mordant mean na say e go help that primary stain wey be crystal violet to stick very well for inside the peptidoglycan cell wall. Now, how you go take do this one? The thing wey go happen na say e get wetin we dey call complexes. So you go form complexes between crystal violet and iodine. Complexes go dey formed between crystal violet and iodine, and those complexes go dey formed inside the cell wall. And as they dey there, you remember say iodine too, the color na like purple. Crystal violet, the color na purple. So they go dey there, they give that bacteria that purple color. Now, after that, the third chemical wey you go add na chemical wey we dey call you add either acetone or ethanol, and we dey call them decolorizer. They call them decolorizer because they go help to wash away that primary stain. The work wey they dey do na to come wash away the primary stain. And the last one wey you go add na chemical wey we dey call safranin or carbonfuchsin. Safranin or carbonfuchsin. This one na the secondary stain. So normally wetin dey happen be say if the primary stain no dey too strong, that acetone wey be the third thing, acetone or ethanol, go don wash away that primary stain, and so the secondary stain wey be your carbonfuchsin or your safranin go fit stay for the body of the microorganism now, or the bacteria. Now, e dey important to understand the science, wetin dey actually happen for this place. Now, wetin dey happen na say if bacteria na gram positive bacteria, that bacteria go get very thick cell wall. Now, if you get very thick cell wall, wetin go happen na say the time wey you put that primary stain wey be crystal violet come add iodine, e go tanda very well for inside the cell membrane. Nothing go fit remove am for there. So if you put the decolorizer no go get the ability to wash away wetin don dey inside that thick peptidoglycan layer, meaning that when you add the fourth chemical wey be safranin, e no go fit enter, e no go see road enter, because the primary stain don dey very well for inside that membrane, because the membrane dey thick. This one na wetin dey happen for gram positive bacteria. But gram negative bacteria get cell membrane, cell wall wey dey thin, very thin. So meaning say the time wey you put the primary stain come put the mordant, they go dey there, but if you come put your ethanol, e go wash away all those things, because the cell wall no dey thick enough to retain that primary stain. So the decolorizer go wash away everything, meaning say the time wey you come put that fourth chemical wey be your safranin, your carbonfuchsin, that one go stay for there. So na that color you go dey see, and all this one na if you don't mount them on top microscope, you can't dey see all these different colors. So this one na the main thing wey dey for inside gram staining. I hope say you understand wetin we don discuss. If you no understand, you fit run the video back 2-3 times to give you good understanding. Abeg, like our content, share our content, subscribe to our channel, and have a wonderful day.